Okay, this is the fourth lecture in the COVID-19 lecture series, and in this session we're going to be looking at the detection of SARS-CoV-2 by various screening and detection methods that can be used to detect either the presence of an ongoing infection or previous infection in patients. So we're going to look through the three main methods of detection of either a current or historic infection, and that is polymerase chain reaction based tests, antibody tests and antigen tests. And to understand how PCR works, we're going to work through a polymerase chain reaction primer design tutorial to illustrate how the technology works. Okay, so you've seen this diagram before. It shows the viral life cycle. It shows the virus attaching to a host cell, and that host cell expresses ACE2, which we've talked about before, and the spike protein is crucial for accessing this host receptor, allows the virus to be internalized. We then get uncoating of the viral this vesicle and also the viral protein and membranous coat, and that releases the free viral RNA. And remember, the virus has got an RNA genome, not a DNA genome. So that RNA is copied within the host cell. That allows translation of that RNA, those RNA copies. So ribosomes bind onto the RNA and translate the various genes within the viral genome to produce all of the various proteins that the virus requires for its life cycle. Those proteins are expressed, they end up in the rough endoplasmic reticulum and Golgi, and the viruses are pieced together and then bud from the cell. So we can detect two different things to detect the presence of active virus, that is the viral RNA, or any one of these viral proteins. So if we can detect either of those, we know that viruses are present. So the viral RNA here is detected by quantitative polymerase chain reaction, and I will only really burden you with polymerase chain reaction, not the quantitative elements to this technique. And this detects the viral RNA. It's the most widely performed test. It's the one that you see on the government dashboards as most commonly used for diagnosis. It has a very high sensitivity and specificity, and we'll come on to what they mean in great detail next week. But sensitivity relates to, out of 100 positive swabs, how many of them would this test detect? And specificity is, out of 100 negative swabs, how many false positives would the test generate? And we know the specificity of this test is about 99.9%, so less than 1 in 1,000 false positives, but we'll talk about that next week. And the sensitivity, well, that's problematic. It all depends on whether the swab has been placed onto a bit of infected tissue or not. So the biggest problem with sensitivity is the swabbing process, because some swabs come out of infected patients with no virus on them. If there's any virus there, this test will almost certainly detect it. So that's the detection of viral RNA. We also have antigen detection. And antigen detection is detection of one or more of these viral proteins. So we can detect specific viral proteins using antibodies, and antibodies, you'll maybe have come across them as part of the immune system. They are proteins that we produce that recognize pathogens, but we also use them in detection systems to detect specific proteins. So if we can get hold of some antibodies that specifically recognize this virus, we can find out whether those antibodies bind to the antigen, which is any one of these proteins. These are less sensitive than the PCR, and they're also less specific. So these tests might be easier to do, but the data that you get out of them is not quite so good. Finally, we have antibody testing, and this is testing whether a patient has previously had an infection and if the patient has had an infection, they should produce their own antibodies to the virus. If we can detect the presence of those antibodies, we know that the patient has been exposed to this virus at some time in the past. Problems associated with this is, like with the antigen testing, we get some false positives. So some people come up positive on this test when they haven't had the virus. We also know that some people who've had the infection do not produce a strong enough antibody response, so we don't detect any antibodies, and that could be as high as 40 or 50% of infected people are antibody negative. Okay, so we're going to start off with polymerase chain reaction, and we're going to cover how this works and then how it is used to detect the SARS-CoV-2 RNA. So polymerase chain reaction is a method of selectively copying and amplifying a very specific region of DNA. Now remember, the virus has got an RNA genome. This test is for DNA, but we can get around that. Now, to amplify a desired sequence, you need to know the target DNA sequence that you want to amplify. And when I say amplify, what I mean is we're starting off with a very small amount of viral RNA on a swab, and we are duplicating a very small region of that genome. Remember, the SARS-CoV-2 genome is about 30,000 nucleotides in length. We will be amplifying maybe just 100 base pairs or 100 nucleotides of that sequence and creating millions and millions of copies of just that small sequence with, just one small part of the genome. So when I say amplify, I mean I'm making millions of copies of that DNA. Now, because it's got an RNA genome, we need to convert that RNA genome to DNA, and we use an enzyme called reverse transcriptase. And what reverse transcriptase does is it can bind to a piece of RNA and basically create a complementary DNA copy of it, and you end up with a DNA-RNA hybrid molecule, which we can separate easily by heating that up, and you end up with a piece of single-stranded DNA, which is a copy of the RNA genome. Now, all we have to do is, and I say all we have to do, we chemically synthesize some short pieces of DNA that are single-stranded, and we call these DNA or PCR primers. They're typically about 16 to 20 bases or nucleotides in length, and they are complementary to the viral genome, or in this case the viral cDNA, at two regions, typically 50 to a couple of hundred base pairs apart. So we've got this 30,000 nucleotide stretch of RNA that's been converted to cDNA, and we're going to bind two pieces of DNA to that in close proximity to each other, and basically the region between those two primers is going to be selectively amplified using a DNA polymerase enzyme, which I'm going to go through. So to allow this technology to be done, we use a DNA polymerase, so an enzyme which extends a DNA chain. It can extend single-stranded DNA and turn it into double-stranded DNA, as long as it has a short stretch of double-stranded DNA to start off on, and that is what our DNA primers will perform. I will illustrate this later. So we provide DNA polymerase, which is heat stable, and this allows the enzyme to be heated up to 94 degrees, and that's crucial for separating the two strands of DNA into single strands, and you'll see that happen. We include a reaction buffer, typically contains some magnesium, salts, and we supply this enzyme with unlimited amounts of deoxynucleoside triphosphate, so the DNTPs, DATP, TTP, CTP and GTP, which are the building blocks of DNA. So we supply the enzyme with primers bound to the complementary DNA. The DNA polymerase will extend those primers and fill in new DNA and create new double-stranded pieces of DNA. And we do all of this on a fancy heating block called the PCR block. Okay, so at this point it's a good idea to click on this link here and watch this very brief animation on how PCR works. So have a click on that on the PowerPoint version of the slides, and then I will start explaining the principles of polymerase chain reaction. So as shown on that animation, during PCR we have our template DNA. Now in this case it's a DNA-RNA hybrid that has been created by this reverse transcriptase enzyme copying the RNA to make a DNA copy of it. During the PCR, what we'll initially do is heat all of this DNA to 94 or 95 degrees, and that will denature the DNA, separating it into two strands. If it's double-stranded DNA, we'll have two single strands. If it's a DNA-RNA hybrid, it will separate that RNA from that DNA. So at 95 degrees, DNA melts. At 60 degrees C, the primers, which are those little bits of DNA that we are supplying the enzyme with to get started, they will anneal to the complementary regions of that target DNA. So they'll be base for base, opposite and antisense to the template DNA. We then heat the reaction up to 72 degrees, and the DNA polymerase has optimal activity at 72 degrees, and it will start moving along that DNA strand, copying that DNA. And as we'll find out on this PCR design tutorial, the two primers are in close proximity of each other, so that's usually within 100 base pairs of each other, and they are what I call pointing towards each other but on opposite strands. And we'll see what I mean when I go through the PCR tutorial. But the result of that, it means that the region between the two primers will be selectively copied by DNA polymerase. The rest of the genome is completely ignored. We've got two primers 100 base pairs apart. The region that encompasses those primers and the region in between is what is going to be selectively amplified. And we repeat this process for about 30 cycles, and we may well end up with millions of copies of DNA just for that 100 base pair region. So before we go through that tutorial, which I will do in a few moments, I'm going to give you a bit of background information about the structure of DNA. Now DNA has two ends to it, and I will repeat this time and time again. It has a 5' end and a 3' end. Now the 5' end of DNA is named because if we look at the structure of a individual nucleotide, this is a deoxyribose, 5-carbon sugar. This is the base, in this case a C, and here we've got a carbon here, and then we've got a phosphate group here. And the backbone of the DNA structure goes phosphate, sugar, phosphate, sugar. Now when DNA is extended, it is always extended from what we call the 3' end. Now if we look at this nucleotide, this is a deoxynucleoside triphosphate which is about to be incorporated into this extending strand here. So the C has paired up with the G. Remember, C goes opposite G, A goes opposite T. Here is the ribose, and the carbons on ribose are numbered from 1 to 5. And this is number 1, number 2, number 3, number 4, and then this one is number 5. Now on carbon number 5, there is a triphosphate group. That's 3 phosphates stuck together. Just like in ATP, adenosine triphosphate. This is a triphosphate group, and it's on the carbon number 5. On the third carbon, so 1, 2, 3, there is an OH group, an oxygen and a hydrogen, and this is really important because the reaction that occurs when a new nucleoside triphosphate is added to DNA is a reaction between the 3' OH on the existing DNA reacting with the triphosphate group on an incoming nucleotide. And that bond creates a new phosphodiester bond, which is part of this phosphate sugar backbone. So the important thing to be aware of is that DNA can only be extended in a 5' to 3' direction. That is, 5' is a start and the DNA polymerase will head towards the 3' end, constantly adding nucleotides at this 3' end as a result of this OH group reacting with this phosphate group. So you need to have extension from the 3' OH from an existing nucleotide. And that is effectively what a primer is doing. It's providing a 3' OH, so it's providing a short stretch of double-stranded DNA that DNA polymerase can hang on to and then react this incoming nucleotide with this reactive oxygen group here to create a new phosphodiester bond. And then the process repeats all the way down the strand. So this is why we say extension is from a 5' to 3' direction only. So this animation is based upon amplifying chromosomal DNA, so we'll just ignore that bit for a moment. But this animation illustrates how PCR reactions get going to produce a selective amplification of DNA. So let's imagine we're adding in some double-stranded DNA to the start of this reaction. Here's the double-stranded DNA. It could be millions of bases of DNA from one end to the other, but we're only interested in amplifying this short region here. So what we do, we heat up this DNA so it becomes single-stranded, and then we, in that heated up DNA, we have some primers which we have supplied to the reaction. And those primers, one will bind to one strand, one will bind to the other strand, and they are effectively pointing towards each other, in that this piece of DNA here has got a 5' and a 3' end, and this primer has got a 5' and a 3' end. And if you remember that DNA can only be extended in a 5' to 3' direction, this end here is the 5' end, and DNA polymerase is going to attach to this end, which is the 3' end. So that is the 3' end, and that will extend, DNA polymerase will extend in that direction. By definition, if this is the 5' end, this is the 3' end of the other strand, because DNA is anti-parallel. Now exactly the same thing is happening on the top strand. Here is our, what we call our reverse primer. This is the forward primer, this is the reverse primer. This is the 5' end, this is the 3' end. DNA polymerase is going to go this way. And if this is the 5' end, then this is the 5' end of the what we call the template strand that it's copying, because DNA is anti-parallel. So we've heated up our DNA, we've cooled it down, our primers are bound, DNA polymerase is copied along here, and DNA polymerase is copied along here until we get to this situation. We've now got two double-stranded bits of DNA where previously we had one. What we do now is heat up this again to 94 degrees and then cool it down, and more primers will bind both to the original template and the newly synthesized piece of DNA from here. So this piece of DNA was created with a reverse primer, and now a forward primer is going to come in, bind to it, and extend in this direction until it reaches the end, and then it will fall off. Similarly here, this piece of DNA, the end of it is defined by the forward primer. The reverse primer is now going to bind, extend all the way along here until it comes to the end and then falls off. So we end up with this situation, 4 pieces of double-stranded DNA. All we do now is reheat that and allow the process to occur, and again we then end up with 8 pieces of DNA. And then we double the amount of DNA every cycle, so we end up with 16, 32, 64, 128, 256, on and on and on, and you can do the maths to find out how long it takes to get to 1 million pieces of DNA. And we can be at that point within about an hour of this reaction going, because each cycle will only take a couple of minutes. Heat, cool, extend takes about 2 minutes. So to summarise, the primers are extended in a 5' to 3' direction. We have 2 primers on opposite strands and they are extended towards each other. This means the region between the 2 primers will be preferentially amplified, and we will basically have a piece of DNA that represents the first base of the forward primer all the way through to effectively the first nucleotide of the reverse primer. And we'll illustrate that on the PCR design tutorial that we're going to do next. Now, these PCR primers that we use will bind elsewhere on the genome, but they might match partly, they might extend a little bit of DNA, but they need to have two primers binding close to each other to get this sort of exponential amplification that we observe in PCR. So if primers bind elsewhere in the genome, it doesn't really matter. It only matters if they bind right next to each other, at 100 base pairs apart. So what we're going to do now, I'm going to finish this video and then we'll do a new video explaining the PCR design tutorial.